Well, hello. Welcome to a Physionic detailed study analysis. Today we're going to be going over cardiovascular disease and how omega-3 fats affect cardiovascular disease. So we'll be going over a number of studies. We'll also be going over side effects and a few different populations of people and how it specifically affects those individuals. If you're not familiar with who I am, my name is Nicholas Verhoeven. I would say I'm a PhD candidate in molecular medicine at this point, because that's what I've been saying for the last few years. But I just recently graduated, so I am now Dr. Nicholas Verhoeven, and with my PhD in molecular medicine. I also hold my master's degree in exercise physiology. But enough about me. Let's go ahead and get into this detailed analysis of multiple studies. If you'd like to skip to the conclusion sections, I'll have a section where you can skip to that's going to give you a rough summary of what we've just covered, but there's a lot of detail that you'll be missing out. So without further ado, let's go ahead and get into this. Okay, so the topics that we'll be covering here are the mechanisms of action, as usual, omega-3 on all-cause mortality, omega-3 on cardiovascular disease mortality. So does it have an effect on reducing cardiovascular disease mortality, as well as some of the side effects, so atrial fibrillation, actually a few others as well. Then if you're part of my Physionic Insiders, I'm going to go into appropriate dosing for omega-3s for the best results, according to all these studies. Omega-3 for people without heart disease, which I think is especially important because we focus so much on people that already have heart disease, which I'll be covering, but I'd like to also create some content and look at people that don't have heart disease and see if omega-3s may still have a benefit, as well as omega-3s compared with statins, so in combination with statins, as well as the types of omega-3s that may matter. So which one may be better than the other, because there's a few different versions of omega-3s. If you're interested in accessing the full version of this video, then just hop on over to the Physionic Insiders, where you'll get access to this one, as well as all my other investigations, all in one place. Okay, let's go into the mechanisms of action. This is a really interesting one. I learned a lot from this one, so I'm really excited to present this to you. So the information is going to be largely coming from study 303. There's no funding or conflicts of interest related to this one, mainly because it's a scientific review. So, there's not going to be a bunch of funding that needs to go into something like that, because they don't have to recruit participants and things of that nature. If you want to know more information on the funding sources and whatnot, just click on the link in the description, and you'll have a downloadable document that tells you more information on the funding sources, as well as gives you the references themselves. Okay, so let's zoom in here. Let's zoom into our cardiomyocytes, which are these heart cells, heart muscle cells. And we're going to see what happens to the cardiomyocytes when we supplement, or at least mechanistically understand what happens when we supplement with omega-3s. So there are actually more than these two versions of omega-3s, but these are the big two that we often discuss. So docosahexaenoic acid and eicosapentaenoic acid. They're also abbreviated as DHA for the docosahexaenoic and EPA for the eicosapentaenoic acid. And all you really need to know is that these are polyunsaturated fats that have a particular chemical kink, as we can see by the illustration here, that occurs at a particular bond site on the chemical structure. Do you need to know about the actual chemistry and all that? No, you don't. So I'm just going to go by it. Just know that omega-3s are polyunsaturated fats of a particular chemical structure. Okay, so these omega-3s enter into the cardiomyocyte, and then what actually happens? Well, what we have to do is we have to zoom into the cell membrane. So we've got the inside of the cell here. We've got the what's called the cytosolic section, or the intracellular section. Then we have the extracellular section, or outside of the cell. So this cell membrane creates a separation between the outside of the cell and the inside of the cell. And that's critical for a number of reasons. I won't go into all of them, but you want to have separation between the two because you have signaling molecules that are found inside the cell that need to be separated from outside of the cell. As one example, you have a membrane potential, so the inside of the cell is far more negative than the outside of the cell. So you want to be able to maintain that membrane potential as well. And that's especially important when you're talking about like heart cells and brain cells, because the cells become activated if they become overly positive inside. So it can lead to aberrant activation. But again, that's a story for a different time. Now, one thing that you should know is that the cell membrane is not just made up of what's known as these phospholipids, where you have this particular structure of fat that makes up the entire lipid. It's actually also inundated with these cholesterol domains. Now, these cholesterol domains are these aggregations of cholesterol molecules. So you may have heard of cholesterol as like LDL or low density lipoproteins. That's not what we're talking about here. We're talking about the actual cholesterol molecule. So the chemical structure of cholesterol, which LDL, the carrying molecule, contains cholesterol, excuse me. It actually has cholesterol inside that LDL particle, but what it's actually depositing is not the particle, it's depositing these cholesterol molecules. So just because there's a lot of carryover between those two, and that can be really confusing for people. So just know that we're talking about the cholesterol molecule itself. So we can have these groupings called cholesterol domains. I believe that if they come in a particular orientation, cholesterol plus sugars plus proteins plus some additional fats, that they're actually called lipid rafts. And lipid rafts can act as anchoring points for different signaling proteins, but we'll get into that. So you can have these sections of the cell that have these cholesterol domains, and that's what I'm going to stick with for the time being. So these cholesterol domains, or you can have these large sections of the cell membrane that don't have any cholesterol domains. So it's not that it's necessary to have these cholesterol domains, but there are certain advantages and in certain instances disadvantages if you have cholesterol domains in areas that you don't want cholesterol domains. So they change generally the membrane fluidity as well. So this cell membrane can become stiffer or it can become more pliable, more flexible. So the cholesterol domains can have an impact on that as well. Okay, so let's talk about omega-3s and how omega-3s have an impact here. So if you have the incorporation of these omega-3 lipids, these fats, then depending on the type of omega-3 that we're talking about, if that's eicosapentaenoic acid or if that's docosahexaenoic acid, so EPA and DHA, each one of them, according to the scientific review and these researchers, has a different effect on the cell membrane. And I'm not going to be able to detail all of them because there's quite a lot. But one of the differences in the effect is that if you have one type become integrated into the cell membrane to a great degree, it can actually block the incorporation of these cholesterol domains. So you actually see you see more of this integration where you have far fewer of these cholesterol domains, these aggregations of cholesterol, than you would in other scenarios where you may not have those omega-3s. Now, to be clear, however, that's not always the case because in certain instances, you actually see more cholesterol domains included with the omega-3 fats. So that's rather confusing. And I don't want you to walk away from this thinking that there's some sort of applicable takeaway like, oh, I need to take it at a certain time or I need to take a certain type just so I can perfectly make my cell membranes proportional. If you start thinking in that level of granularity, it's going to get bonkers because cell physiology and molecular physiology is it's way too complex to try to think about those kinds of minutia. So what we're trying to get at is, should you take omega-3s or should you not take omega-3s, and let your cells figure out what it needs to figure out to make things as appropriately healthy as it needs. So yes, there are certain omega-3s that will increase the density, and I believe the increase in density is due to the DHA. So DHA is the one that typically increases the number of cholesterol domains, and EPA tends to shun or block the integration of these cholesterol domains. All right, now let's talk about some of the negatives of not having omega-3s. And I'd like to remind you that this is based off of mechanisms. We're not looking at clinical data yet. So what I'm presenting here is just what the researchers point out, but we need to actually see the data for ourselves. So remain unconvinced. That's my point. Just remain unconvinced, because mechanisms are interesting, they're cool, but they don't actually tell you about, does it actually improve cardiovascular disease risk? Okay, my soapbox out of the way. Now let's talk about what happens when you don't have these omega-3s inside the cell membrane here. So you can see, I usually denote this as kind of the yellowish if it has omega-3s. And here we see that there are no omega-3s, or let's just say low levels of omega-3s. So we think of the cell membrane as this just static structure that's just separating out the extracellular from the intracellular, which is what I talked about earlier, or allows for cell membrane dynamics like the membrane potential and things of that nature. But in reality, the cell membrane itself can be a signaling cascade, can be a propagation of signaling within the cell. What do I mean by that? Well, remember, there are these phospholipids that make up the cell membrane, and the phospholipids can actually attract, depending on the type of phospholipid, can attract different proteins to the cell membrane. One of those proteins is called phospholipase. And phospholipase, when it interacts with certain phospholipids, it can lead to the production of a molecule called arachidonic acid. So, and I want you to keep in mind that this is important, because in a few slides, I'm going to show you the exact same thing, but the proportion of each ingredient, so the amount of arachidonic acid, is going to decrease. And here we see that there's a good amount of arachidonic acid being produced by this phospholipase that interacts with these phospholipids. And then what goes on from there is the arachidonic acid gets acted on by a family of enzymes called oxygenase enzymes. These oxygenase enzymes then produce this family of molecules called eicosanoids. So the eicosanoids then get released from, in this example, we're talking about cardiomyocytes, remember, so they get released from the cell, which then can bind to immune cells and then recruit immune cells to this area. And the reason why that's potentially problematic is because immune cells, when they're in their activated state, can lead to a lot of destruction around them, because they're in this pro-inflammatory state. So they, for example, they'll start producing a lot more oxidative stress and release those damaging molecules, those free radicals, into the environment, into the nearby cellular environment, the extracellular space, and start damaging the molecules and the cells around them. Now, it's not that your immune system is just like dying to kill you or anything like that, but when it interacts with pro-inflammatory molecules like eicosanoids of different shapes and sizes, then it can lead to this increase in pro-inflammation. Okay, so one of the mechanisms by which this occurs, and this is in endothelial cells. So at this point now we're moving away from necessarily the heart, and we're looking at the blood vessels themselves. So the blood vessels, think of it like a really dynamic tube, and the blood vessels have this lining of cells that essentially control the tube and control the amount of blood pressure that's found in any one region of the blood vessel. And those cells are called endothelial cells. So another thing that eicosanoids can do is when they get released, and this is not specific to just the cardiomyocytes that I was showing you, I was just using it as an example. This actually occurs in many of the different cells of our body. So these eicosanoids get released, and then they can bind to a receptor called the thromboxane receptor. And then from there, the thromboxane receptor can activate a signaling cascade called ROCK. So there's this protein called ROCK that's found inside the cell. Remember, this is the intracellular section, or the cytosolic section of the cell. Now, there's way more complexity to this than what I'm showing here. This is a massively reduced version of what would really happen. Yes, you would get the binding of icosanoid to the receptor would lead to, there would be a physical change in the thromboxane receptor called a conformational change. And then from there, that would attract different proteins, among which ROCK is one of them, to that region. And then once it binds to the receptor, ROCK gets activated, and then it has a number of proteins. So these arrows are essentially eliminating all those proteins that ROCK would then interact with to activate those. But ultimately, there's a signaling cascade of many proteins that get activated by ROCK, which leads to greater focal adhesion proteins. I'll show you what that is on the next slide, and endothelial morphology changes. Again, I'll show you what that looks like. But those are two of the things that can change. Just to be 100% clear, I'm showing you a lot of mechanisms here, but even this is not all-encompassing. Okay, so ROCK gets activated, you get greater focal adhesion proteins. What does that mean? Well, focal adhesion proteins, and this is the blood vessel I was talking about earlier. So you've got, if you were to cut your blood vessel in half and just kind of look on the side of it, as if you're looking through the side of a hose, and you've got the endothelial cells here, and then you've got different intima thickness. You have smooth muscle cells and things of that nature in here. I'll be showing you a little bit more imagery on that a little bit later on. But for now, just know that you have this single line of endothelial cells that are in contact with what's known as the lumen, or the opening of the blood vessel. And this is where, the red blood cells are moving through. There's different proteins like albumin. You have immune cells. All these different things are circulating through. So every time you pump with your heart, you've got blood flow that continues to move, plasma flow that continues to move. And that's what's happening here. Okay, but we're not as interested in that for the time being. What we're interested in is in these focal adhesion proteins. So focal adhesion proteins are these proteins that can be expressed on the surface of the endothelial cell. Let me go back here. So the focal adhesion protein would be expressed and then get transported up to the cell membrane here and integrate into the cell membrane. And that's what we see here. It's a little bit exaggerated because they wouldn't be quite this large, but the point is that you have the cell membrane and the focal adhesion protein is stuck in it. And what happens is that when immune cells float by and they can interact with this focal adhesion protein, which then allows them to essentially drive into this area. So they attach to the focal adhesion protein. So technically they roll, and as they roll, they slow down, and then they bind to the focal adhesion protein and cell adhesion proteins in general. And then they can invade past the endothelial cell to get into this sublayer over here, the subendothelial layer. So under endothelial, these cells layer. So under that layer, which is problematic if you have plaque buildup, because as plaque starts to build up, the inflammatory process is a major contributor to the continuation of the progression of that plaque. So atherosclerosis, plaque buildup, becomes worse and worse as you have more and more of these immune cells that are invading in this area in a pro-inflammatory manner. So more focal adhesion proteins leads to more of these immune cells that can then find their way into the subendothelial space and can ultimately eventually lead to a more progressive plaque formation. So clearly not a great thing. And that's where these icosanoids can unfortunately contribute. The other thing that they can do is that icosanoids can bind to the endothelial cells and lead to changes in its morphology. So I said endothelial morphology changes, and that's what we see here. So we can see the cell can actually contract in on itself and essentially shorten itself and make itself smaller. So what happens in that situation, now you have greater gaps by which you can have different LDL particles or ApoB containing particles, immune cells, and a bunch of other things that can then be exposed or more easily exposed to the subendothelium, and they can move through more easily. On top of that, you can also have the exposure of different proteins, and the exposure of different proteins from the subendothelium, for example, like collagen, can also activate the platelets. So you've got these platelets that flow through the bloodstream, and when they interact with certain molecules, they can become activated. When they become activated, they can aggregate and essentially create a clot in that region. Now, of course, if that continues to grow, then that can be problematic in its own right. So there's a number of problems with leading to unwanted changes in the endothelial morphology, the actual shape of the cell. Another thing that happens with eicosanoids is once they bind to the thromboxane receptor, again, they bind to other receptors as well. I'm just focusing on one. So the thromboxane receptor, not only does it function through ROCK, but it also independent of ROCK, because technically if everything ran through ROCK, we could eliminate the impact of these eicosanoids by maybe using a drug that targets ROCK and just lowers its expression or lowers its activity or something along those lines. But that's not the case. Unfortunately, eicosanoids have there's a pan effect where it affects many different cell types as well as cell receptors. Okay, so eicosanoids bind to this thromboxane receptor. It does not affect ROCK in this situation. I mean, technically it probably would, but we're talking about a mechanism that's been identified independent of ROCK. And it leads to a signaling cascade. Again, I've removed that signaling cascade. Just know that there's a signaling cascade that comes from the receptor down to the nucleus. Inside the nucleus, you have these proteins called transcription factors that will then move from the cytosolic region, so that's this whole region, so inside the cell, but outside of the nucleus, will then move from the cytosolic region into the nucleus and bind to particular genes. Now, those genes, one of those genes is called the COX gene, so that's short for cyclooxygenase. And this cyclooxygenase enzyme, or this gene, I should say, then gets transcribed and translated to the COX enzyme, which is the actual folded protein. It's the actual functional protein. Now, this COX enzyme is responsible for also producing eicosanoids. So it is one of these oxygenase enzyme, part of that family. And what it actually specifically produces is the molecule called prostaglandin. So prostaglandins then increase the inflammatory cascade that I was talking about, and it attracts more of these inflammatory cells. So obviously, this whole process is just another way by which eicosanoids can have a repeat, kind of a futile cycle of just continuously increasing. Now, your body has ways, your cells have ways of interrupting that system so that you're not constantly producing more of these eicosanoids. But we're focused on omega-3. So I've talked to you about the bad. Now let's talk a little bit about the good of what's the mechanisms been implicated in this. So omega-3s have been shown to directly impact COX enzymes. So even though it gets produced, it actually gets blocked by omega-3 consumption. So if you have free omega-3s, or even if they're incorporated in the cell membrane, you have less of the activity of COX. So therefore, then you have eicosanoids then decrease in their amount, their concentration. Okay. Now, I mentioned that you should pay attention to the amount of arachidonic acid and eicosanoids. Let me go back and remind you here. Look at the cell membrane. There's no DHA, EPA added. There's no omega-3s added. You can see that there's high levels of arachidonic acid. By the way, this isn't evidence. I just want to be clear. This is me just giving you information based on the scientific review. Sometimes people think that if a person talks confidently, then that must absolutely mean that there's evidence, which is just like a side note that I think about sometimes. You see these like presentations on social media of like people just talking very confidently. Look, there's no DHA, and there's high arachidonic. That's not evidence. That's not actual data. So I just want to point that out. Just a side note. I'll get back to on topic though. We'll be looking at actual evidence of some of the stuff that I'm going to be talking about. Okay, so just note that here, based on the review, that there's no DHA, no omega-3s found in the cell membrane. There's high levels of arachidonic acid. There's high levels of eicosanoids. Now we go back and now look at the cell membrane. Ooh, there's a lot of omega-3s included in the cell membrane. You still have the binding of phospholipase, but there's less binding of phospholipase to these phospholipids because it's actually impacted by the structure of the phospholipid. I suppose I should have explained that the phospholipid is partly made up of fatty acids, of the fats that omega-3s are. So if you have a phospholipid that's made up of other polyunsaturated fats or other saturated fats, then that would attract more of the phospholipase. And if it has more of the omega-3 incorporated in that phospholipid, so inside this structure, so you see this, how this one is kinked slightly, that's the idea, that this chemical structure slightly changes in such a way that phospholipase is now no longer attracted to the cell membrane and therefore has less of an opportunity to bind and produce arachidonic acid. Now, additionally, just by sheer concentration, if omega-3s take up more space in the cell membrane, that de-emphasizes the amount or the concentration of other phospholipids, which then leads to, again, lower arachidonic acid, which, even if you have normal high levels of these oxygenase enzymes, they don't have much substrate to work with, and therefore they're going to produce less eicosanoid production. So that's another mechanism by which omega-3s can have an impact. Now, it's not just defense, though. Interestingly enough, this is something that I learned when I was reading about this, when I was analyzing some of the studies, is that it's not just defense, it's actually also offense. So omega-3s can lead to the production of, think of them like the angel molecule to the eicosanoids. And I'm not even sure if eicosanoids are necessarily always bad, but in this context, they tend to be pretty negative. But omega-3s can be produced or can lead to the production of what's known as specialized pro-resolvin mediators. So SPMs. So let's see what SPMs do. And they're in green, so they must be good. Okay, so SPMs can be released out of the endothelial cells, and they actually have a number of effects. Well, one, they can actually bind the endothelial cell itself, so that's known as an autocrine action, meaning that it gets released by an endothelial cell and then it binds to an endothelial cell. The other thing that it can do is actually bind inside the subendothelial layer. Remember that there are these smooth muscle cells that are found inside this layer, and it can bind to that and modulate their activity as well. So how does it do that? Well, here we've got our cell membrane. This is a kind of a generic cell membrane. We've got the exterior section of the cell. Remember, that's called the extracellular area. And here we've got the intracellular or cytosolic section. Here we've got a particular type of receptor called a GPCR, so a G-coupled protein receptor. It kind of gets jumbled up, but the same thing. So GPCR, this SPM molecule, this mediator molecule will bind to the exterior section, which will lead to, again, that conformational change, which will lead to these different molecules interacting in a particular way. So there's a grouping of molecules called G proteins that interact in a particular way that leads to further internal signaling inside of the cell. Now, I'm not going to be showing you all that. So again, these arrows represent a lot of skipping of different molecular interactions, because otherwise this presentation is going to be like 5 hours long. And we see that there's reduced cell migration. So that's one aspect. If I can go back here real quick. When you have more advanced atherosclerosis or plaque buildup, one of the problems is that, or I shouldn't say problems, but one of the symptoms of it is that once you have this plaque that kind of builds up over here and pushes the endothelial cells outwards into this lumen and then restricting the amount of space for blood to flow through, there's an unstable section near this whole area that's starting to protrude is very unstable. So your cells will migrate, specifically your smooth muscle cells will migrate and create a kind of a cap. It's part of a cap, I should say. It's not the whole cap, but will create a cap over the actual atherosclerotic plaque. And this SPM will reduce the cell migration. Now, there's negatives to the actual migration because it means that you're progressing forward in the atherosclerosis process. Additionally, reduced oxidative stress. So one major factor for atherosclerosis is also oxidative stress. So you need to have the oxidation of these ApoB proteins that are found on some of these lipoproteins. I have content on all that, so I'm not going to go into too much more detail on that. But the point is that if you can reduce the oxidative stress, not only are you affecting the external environment, so not only are you affecting all this area, especially the plaque buildup that may start to build up from the ApoB containing proteins then getting stuck down here. Now, once those become oxidized, then you have a lot more problems. You have more atherosclerosis progression. You also have the breaking of the lipoproteins. You can have the release of the free cholesterol, things of that nature, which can lead to even more inflammatory cascade. There's a lot to it. So not only does reduced oxidative stress have an effect on that, but it also, and reducing it, but also it can have an effect on the cells themselves. So now your cells are less likely to die. Your cells are more likely to become robustly inactivated against pro-inflammation. So there's a number of benefits of reducing oxidative stress and also reducing cytokine production. So in a way, you can think of the eicosanoids, the red molecules that I showed you earlier, when they were attracting the immune cells. Those are called chemokines. So they're kind of like a specific type of cytokine or a very close relative to the cytokine. And cytokines and chemokines, they attract immune cells. So they can change the profile of the immune cell to become more pro-inflammatory, which, as I explained earlier, has a bunch of negative consequences. And if you can reduce the amount of these cytokines that get released from the endothelial cells, then that means you have less of this activation of the inflammatory cascade. So reducing that as well. Now, this is kind of pitiful, to be honest with you. And if this were a more mechanistic presentation, I would walk you through exactly how it does that, molecule by molecule, what it's interacting with. But for now, this is what we're going to have to stick with. So these three benefits, and surely there are others as well, but these three are really massive. And then finally, just real quick, another factor that I actually wasn't as familiar with, and is really counterintuitive, to be honest with you, because typically polyunsaturated fats are more oxidizable, meaning that free radicals or damaging molecules, reactive oxygen species, can interact with polyunsaturated fats more easily and lead to what's known as lipid peroxidation. So lipid peroxidation is basically damaged fat molecules. So if you have these reactive oxygen species that interact with the cell membrane, then that would lead to damage to the cell membrane because it's ripping away and changing the chemical structure of the cell membrane to be different from what it was supposed to be. Okay, however, this scientific review also mentions that free omega-3s can actually interact with free radicals, reactive oxygen species, and neutralize them. So they can act as antioxidants as well, which I found really, really fascinating and thought I'd share that as well. Okay, folks, let's go into the results. Now we can actually talk some real data. So the first thing that we're going to be looking at is omega-3s and their impact. So omega-3 supplementation on all-cause mortality. So if that's death from cancer, that's death from cardiovascular disease, if that's death from kidney failure, everything that kills you, that is included in this analysis. For the first one, we're going to be looking at study 301, which has public funding. Again, if you want more information on the studies and everything, it's linked. I have a document that has more information on everything. But it's public funding, which means that it's typically funded by some government agency. So public funding as in it came from taxpayer dollars. Here's the design on this meta-analysis. So it includes 14 studies. They go through a rigorous process, identification, screening process, because let me go back here real quick. There's a number of researchers. So they usually have maybe 3 researchers that do this independently, and then they come together at a certain point and then agree, we should be including this study, we should not be including this study. They essentially have to come to a consensus, and they have a different inclusion and exclusion criteria. So if they only want to include randomized controlled trials, then they're obviously going to exclude all animal studies, for example. So they ended up with 14 studies in total. The other thing that's important to look at whenever we have an opportunity is to look at bias assessment. So another thing that researchers will do is they open up each one of those 14 studies. We can actually see those studies here, and they'll open them up and run through these standardized questions that they have to answer, and they've got kind of a grading system that they have to go through. And what we see here, this is actually a little bit more detailed than the usual one that I see. It's usually low, unclear, or high. I've never seen critical before, so that's interesting. Anyway, so we've got the different areas, the different criteria that they're looking at. This is extremely standard. It's part of a Cochrane risk bias assessment, or part of the grade risk assessment. But the point is that you want most of these, if not all of them, to be green. And if you have certain studies that are green all the way across, that means there's very low risk of bias in that study, which is fantastic. That's exactly what we want to see. And I got to say, when this is looking at each study individually, and this is looking at everything put together. So a lot of this is low risk of bias. I know there's a few here that are high risk of bias. That doesn't necessarily mean that the study is bad. It just means that there's a higher chance that there may be some methodological issues with that study that may influence the results. And sometimes studies, these meta-analyses, will do something called a sensitivity analysis, where they do a removal of those high risk of bias studies to see if that changes the results of the overall analysis. Anyway, we don't need to get into that. But the point is, here, I'm pretty impressed. I have to say that this looks really good. This looks actually really fantastic. So a lot of them are low risk of bias, which indicates that there's low risk or low probability that we're going to run into any sort of methodological issues that may confound the results that we're going to be looking at. Another thing to look at is a funnel plot or a pyramid plot for looking at risk and looking at bias. I guess I should say it's more bias related. And the way to read this, I actually go over this in my course, but the way to read this is that, well, there's different ways of the way that they show this. I'll just give you a simplified version. So the results land on this line here, and these are all the studies that are included. Each dot represents a particular study. And what they're trying to show is that if everything lands somewhere within this white section, that indicates it's not statistically significant, meaning that there's likely no difference between these studies, or no really apparent difference between these studies in terms of some confounding variable, some factor that may strongly distinguish this study from all the other studies. So we can see that most of the studies fall in this white area. Again, that's not too surprising, especially when you have such a low risk of bias. However, you can have studies that fall outside of this white triangle. And then usually what the researchers will do is they'll identify those studies, which is, I suppose, just one study in this situation, and they'll remove that from the analysis and report the data with the study and without the study to show if there are any differences. Or they might just say, they might do it and then just tell you, hey, there's no difference between the two, so we just went with the one that included it or excluded it, however they want to do it. So if it falls anywhere in these gray areas, it indicates that there is a difference, a significant difference from the rest of the analysis. Now, how to translate that, you could think of it like, is there something inherently different about this study? So, for example, if all the other studies are done in Europeans, for example, and they're all the studies are done in, let's say France and Spain and England and Germany, and then this one study is done in Vietnam, is there something about the Vietnamese population or society or culture or something that could potentially lead to this difference in the effect? And obviously we don't know that, but you always have to consider it could be because the population is different. It could be because the sample size is too small. You don't have enough people in the the study itself. There's all kinds of different reasons. And this kind of analysis, it doesn't tell you, oh, this is the reason why. It just tells you, hey, this study is quite a bit different from the other studies. So it may be one that you may want to consider just cutting out and seeing how that affects the results. Hopefully that's somewhat clear. Okay, so here we've got the exact same thing. So I'm just showing it to you again. But here we're actually looking at the effects. So the effects on total mortality, or all-cause mortality, I suppose I should say. Okay, so like I mentioned, this is the kind of stuff that I cover in my course on how to read studies. So if you're interested in taking that course, I actually walk you through step-by-step how to read meta-analyses. I walk you through all the details. I go into the statistics and all that. The course actually only takes about 5 hours to go through. So if you have no clue how to do anything related to this, it takes you from being a complete beginner to being proficient, and it gives you a step-by-step guide. And on top of that, it teaches you how to apply this kind of information to your life and what to look out for. So if you're interested, it's called the Health Autonomy course. It's actually in the description box, but no pressure. Let's go ahead and walk through this a little bit. I'm going to give you a little bit of a taste on how to read this. So on the left side here, we've got all the studies. So these are the studies. We've got the total number of participants in each study for the omega-3 versus the control. And usually the control in these situations is some form of placebo, some sort of standard of care that the omega-3 group also receives. So that's usually a pretty tight control, which is nice. The events here is the number of people that have, died, because we're looking at all-cause mortality. So this is the number of people that have died, and this is the number of total participants in the overall analysis. Then I'm not even going to walk you through all this, like weighted on different like the weighting of different studies and stuff. It's stuff I cover in the course, but I think we should probably focus on the actual results so that we can get through this analysis more quickly. Okay, so the one here indicates that there is no benefit, no effect essentially of omega-3 supplementation on all-cause mortality. If it moves to the left, that indicates that there's a reduced risk of all-cause mortality with omega-3 supplementation. And if it moves to the right, that means that there's an increased risk of all-cause mortality with omega-3 supplementation. And all of these single, squares and lines and whatnot, they're averages and confidence intervals. Again, things that I'm not going to go too much into right now, but they represent each one of these studies. So each one of these studies has a representative little image which represents the results of that study compared against the control. So if we average all these studies together, we end up with these diamonds at the bottom here. So let's focus on the red one here. So this red one is the one that you can really focus in on and see, where does it fall on this line? Does it fall to the left? Does it fall to the right? Or does it fall right smack dab in the middle? Now, as I've said before, we can't base study analysis just based on our eyesight and just be like, yeah, I think that seems like it's more to the left, so I'll just say it's more to the left. We actually have statistical tests that look at that as well. And we've got two different models here. So they've got two different diamonds here. Again, those are things that I cover in the course. It's based on the heterogeneity, and it's kind of nice that they actually report both, just to give you an indication of like, hey, if we use this statistical model or if we use this other statistical model, we would get similar results, or we would get different results, which I think more information often is better. And I'll go ahead and tell you that they don't report the p-value here, the statistical test for the, they do right here actually, but only for the fixed effects model. And the reality is, because I've memorized the p-values for all these, the reality is that there's no effect. So even though it seems like it's slightly moving towards the left, indicating a positive effect of omega-3s, the reality is that statistically speaking, there is likely no effect. And it's because the p-value is well above 0.05. It's 0.35. And the relative risk is just 0.98 with some confidence intervals all over the place. So the answer is that omega-3 fats do not impact all-cause mortality. Next, let's look at study 302, which I could not find the funding source on this one. So you'll, it's not reported as far as I could tell. Okay, study 302. We're still on all-cause mortality, just to be clear. Here we've got the background on it. So they included 28 randomized control trials. In this one, I think the last one was 14. So we have even more data for this analysis, which is great. Okay, here we are. It's the same story. I'm not going to walk you through how to read this again. I'm just going to start blasting through this data, but I do want to show it to you regardless. So here we've got that line. Again, if it moves to the left, that's a good sign. Here we've got this teeny tiny diamond down here, but the reality is the p-value is actually very similar to the other analysis. So even with more studies, this further extends our confidence that omega-3s do not impact all-cause mortality. Now, if you're confused by this p-value over here, that's actually in relation to the heterogeneity. And for anybody who has taken my course, they used a random effects model for this, which is good because they have some level of heterogeneity. So anyway, consistency across the board. Next, let's go over study 304. This is public funding. And here we've got 15 studies included, and surely there's going to be a good amount of overlap between these analyses. I think the one that has 20 something obviously is going to have a lot more studies, but just wanted to point that out. And here we have the data. What's a little bit unique here is that they do some subgroup analyses indicating that, which means that they split them up by the dietary supplement, the omega-3 ethyl esters, and EPA ethyl esters. So EPA is not the government agency. It's not the Environmental Protection Agency. It's the eicosapentaenoic acid, which is one of the omega-3s that we talked about, as well as this type of omega-3. So they do a comparison between them so that you can look at each one of these diamonds if they just consider this grouping of data or just consider this grouping of data. And down here is the overall effect, which indicates all of this data combined. And I can go ahead and tell you that the results again were confirmed, that there was no statistically significant benefit. And then of course, you can look at each one of these. You might look at this one, for example, and say, oh, well, this one seems like it's moving to the right. So keep in mind, it's only one study. So, you can see that some of these move way off to the left and some of them move way off to the right. So looking at one study in a meta-analysis and then making a conclusion off that is probably not the appropriate way to go about things. So my conclusion off this is that it confirms the previous data, but no matter which version that you look at, there's no benefit towards all-cause mortality. All right, ladies and gentlemen, let's get into the main event. I need like the announcer voice. Oh, ladies and gentlemen, like the boxing announcer. Okay, I'll stop. Omega-3 on cardiovascular disease. Let's look at this. So first, we're going to be looking at study 301. And if you want more details on how to read these graphs and whatnot, just go back to my all-cause mortality. The first data section there is the one that starts covering or covers in more detail exactly how to read this, so you can get a little bit more of an education. I've got that in the timestamp so that you can figure out where I start talking about it more easily. Okay, so we're looking at 3 different metrics. We're looking at MACE, so that's the different cardiovascular events, major adverse cardiovascular events. Took me a second to get there. Cardiovascular death, so that's the events. That's not necessarily death. This is death. And then this is myocardial infarction, so heart attack. And again, just as a reminder, if it moves to the left, that's a good thing for omega-3 fats, but we can't rely on just our eyes. We also have to look at the statistics. And it turns out the answer is yes, it is statistically significant. There is a benefit against MACE in lowering the risk of MACE. It's not huge. It's a small effect, but there is evidence indicating that it is a benefit. Now, for cardiovascular disease, again, it is statistically significant, indicating that there is a benefit. And again, for MI, myocardial infarction, heart attack, there is again statistically significant effect, indicating that across all three metrics, which sometimes you can have some metrics that don't show an effect. That's because how do I explain this? The way that it's measured is less sensitive than the other measures. So just to give you an example, cardiovascular death, you need the person to die. You can't just which is tragic, but you need, it sounds like, anyway, you need the person to die, as opposed to like MACE, they can have a cardiovascular event, but not die. So you're going to have a lot of people that survive their cardiovascular events. So that means the people that actually die is much smaller than the people that actually have an event. So you have a lot more data to pick through for the events than you do for the death, which lowers the power of the analysis. Again, stuff that I cover in the course, but it's incredibly consistent. So cool. We see that omega-3s are beneficial. All right, next, let's go into study 302. As a reminder, if you need to learn how to read this kind of in a basic sense, then hop on over to the all-cause mortality section where I discuss that a little bit more. Here we're looking at MACE again. So that's, again, major adverse cardiovascular events. And we see that it is statistically significant, indicating that there is a benefit of omega-3s. Here we are looking at, let's see here, cardiac death. And cardiac death is also affected and reduced by omega-3 consumption. Now, just to be clear, these effects are not huge. They're small effects, but they are still a benefit. And we're also looking at myocardial infarction, if I remember correctly. Yes. Now, this one does not technically reach statistical significance. It approaches it. So maybe if they'd had more data included, that they probably would have had a statistical effect. However, as it stands, it's just some people hate it when you say it this way, but it's trending towards being statistically significant. But I think that in considering all the other data that we've looked at so far, I feel pretty comfortable in saying that there's probably an effect here, even though we're not detecting one. If you disagree, that's perfectly fine. Now, to be clear, I wanted to also show this. So this is the first one looking at stroke. So this is a problem that happens in the brain due to cardiovascular disease. And I'd like to point out here that there's actually no benefit towards stroke. So other types of cardiovascular disease, which are related to basically the rest of the body, like heart attacks or major adverse cardiovascular events, those seem to be improved by omega-3. However, stroke is not, which I find really interesting, and I actually don't know why. If you have some thoughts as to why that might be the case, please let me know. Okay, study 304. Here we are. We've got these different breakups like we did last time, and in this situation, we're looking at MACE. Now, again, this is a combination of everything together into this diamond over here, and they don't report the p-value, if I remember correctly. Yeah, I don't think they're reporting the p-value, but I can tell you off of what the researchers point out in the actual text themselves, because they do have to report the p-value. They do indicate it is statistically significant. So this is not the p-value for the overall effect. It's the p-value for the heterogeneity, just so you're aware of the difference between those two. But that, of course, is considering all the studies grouped together. Probably if you were to look at each one of these, like dietary supplements, just dietary supplements, it probably would not indicate an effect. As a matter of fact, it's almost certain. And maybe this one might show an effect, but again, you're only looking at 2 studies, but at least you're looking at more than one study, like unlike this one where you're only looking at one study in a meta-analysis. All right, next we're looking at myocardial infarction. So that's heart attack. And again, I can tell you that it is statistically significant. And here we're seeing a little bit greater relative risk reduction than what we've seen in the past. So that's pretty encouraging. But overall, we're seeing a lot of consistency, which is great. Okay, I wanted to add this one in here as well. This is looking at heart failure. Heart failure is an interesting one because we're looking at the stopping of the heart. but that doesn't necessarily mean it's from cardiovascular disease in the sense of like plaque formation. It can be due to, for example, congenital heart failure. So that can be due to genetics, or it can also be due to, yes, obesity can contribute to it, but it can be due to the muscle hypertrophying. So it's essentially enlarging to a point where it can't pump anymore. So the muscle gets in the way of itself, essentially, of the heart. So omega-3s do not seem to be a benefit in relation to heart failure. And that's probably because we're looking at more than just the cardiovascular disease or the plaque reasons for heart disease. We're looking at, or heart failure, I should say, we're looking at overall heart failure. And some of that is going to be affected by non omega 3 or non plaque related disease. So things that are not going to be modulated by omega-3s being incorporated in the cell membrane and things of that nature. So but I want to, in full disclosure, I just want to show you that specifically for heart failure, it does not seem to have a benefit. Okay, and again, here we find ourselves looking at stroke. And again, the results are no effect. So this is across 2 analyses now. We see that stroke is unaffected by omega-3 consumption. If you're familiar with relative risk, you might see that this increases. That is not statistically significant. So we just assume that there's likely no effect. Okay, now we're talking about cardiovascular death here. And it is not statistically significant, if I remember correctly. I believe that the EPA ethyl esters was considered statistically significant, but as I mentioned, this is only one study. So in a meta-analysis, okay, we can indicate this study indicates that there's a benefit, but overall, if I remember correctly, statistically speaking, no effect on cardiovascular death. And that might be because of the reason that I mentioned before, that with the power, which you can go back to the other measure on cardiovascular death for that explanation. Okay, so we've talked about the benefits. I think we can roughly say that omega-3s are a benefit for cardiovascular disease. Risk, lowering risk. However, are there side effects? And this is something that the researchers also looked at. So side effects, we're looking at atrial fibrillation as well as a few others as well. Okay, so we're continuing on with the study from that we just went over. So we're looking at study 304 in this situation. And here I can go and tell you that statistically speaking, there was a robust statistically significant effect indicating that it didn't really matter. Omega-3s did increase the risk of atrial fibrillation. So if you're not familiar with what atrial fibrillation is, it's a type of arrhythmia or a type of incorrect rhythm of your heart. We'll just call it that. However, what's interesting is that you should also be aware that just because you have atrial fibrillation, what we're also interested in is what's the end outcome? Do you see worse cardiovascular disease? Do you see increases in death? Things of that nature. And so far, the answer has been no, pretty stably no. However, if this scares you, if atrial fibrillation is something that might scare you, then I certainly understand, because I mean, nobody really wants to have an arrhythmia in their heart, and it's not like it would necessarily happen all the time or anything like that. And of course, it's going to depend from person to person, just like in any situation. We're talking about averages here, so it's something to consider. But atrial fibrillation does seem to be increased, and it's maybe something that I can look into in more detail in the future. But for the time being, atrial fibrillation is increased with omega-3 supplementation. Okay, now let me walk you through here. A here is gastrointestinal issues, so like discomfort. B is bleeding disorder, so an increased risk of bleeding. C is cancer risk. A, as much as this seems to move towards the right, it is not statistically significant. That's probably because these are just so massively all over the place. So there's no, and keep in mind, just because it's not statistically significant doesn't mean that there's no effect. It's possible that they just don't have enough power in the analysis to detect an effect. So I think the jury's still out on gastrointestinal disorders or like discomfort. But as it stands, we have no clear evidence that omega-3s increase gastrointestinal disorder. Next, bleeding disorders. So increased risk of bleeding. The overall effect, there is no statistically significant effect. However, EPA ethyl esters between these two studies indicates there is an increased statistically or increased risk. So statistically, and it was statistically significant. So these other versions of omega-3s do not, highly likely do not increase risk of bleeding, but these ethyl ester versions, based on two studies, do indicate there is an increased risk of bleeding disorders. So something to consider, something I wanted to report. C, cancer, across the board, there was no increased risk. Actually, now that I look at it, it's kind of funny that they put EPA ethyl esters down here and then they put it up here, maybe because they want to accentuate it. I don't know, it's kind of odd that they would switch the places, but because you get used to it, right? Dietary supplements was up here, whatever, it doesn't matter. The point is that there's no increased risk of cancer with omega-3s. Didn't matter which type that you looked at. Okay, so unless you're going to be continuing on for the insider version of this, where I'm going to be talking about appropriate dosing for omega-3s for best results, which might also reduce the risk of arrhythmias. There's no clear evidence on that, but I'll be touching on that as well. So appropriate dosing of omega-3s for the best results. Omega-3s for people without heart disease. That's if you just want to see it as a preventative, is it effective? And omega-3s compared with statins, so with statins. If you're interested in that, then certainly join the Physionic Insiders, and we'll be continuing on with this investigation. But for everybody stopping here, what we have, what's the overall result? Should be a space here. Omega-3 supplementation does not reduce all-cause mortality. We have pretty good data on that at this point. Maybe if you increase the studies like so much, then maybe you might be able to tease out a difference, but it's probably pretty negligible. So the effect is that there's no effect on all-cause mortality. However, and there's reasons that I explained for that. However, omega-3 supplementation does improve cardiovascular disease risk across multiple metrics. That was across cardiovascular death, which was, one said yes, one said no. Myocardial infarction, heart attack, that did improve. And finally, stroke did not improve. And MACE, or major adverse cardiovascular events, did improve. So across multiple metrics, there was improvement. In addition, omega-3 supplementation may increase the atrial fibrillation risk. Generally, that's what we talked about, as well as bleeding disorder risk in this specific type of omega-3. Overall, omega-3s are likely a benefit to the heart and overall cardiovascular health. And the evidence was pretty consistent across the board on that. So that's how I would interpret that data. Okay, that's what I've got for you. Hopefully this was informative. Thanks for sticking it out with me, and I'll catch you in the next one. Bye.